Good morning, children. Welcome to Biology Reward, and today we are discussing the class 12th biology practical. And today the practical aim is to study the effect of different temperature on the activity of salivary amylase on starch. So this is the very good experiment, children, and practical board examination of the CBSE class 12th examination. This is a major experiment. So we have to discuss today to study the effect of different temperature on the activity of salivary amylase on starch. So we will see that how the salivary amylase is acting on the starch. So for this experiment, what we require, our basic requirement: test tube, iodine solution, starch powder, salivary amylase, burner, thermometer, etc. So many things we require for this experiment. Now procedure. For the procedure, preparation of 1% of starch solution. Now we will discuss that how to prepare. Preparation of 1% of sodium chloride solution. Collection of saliva, and we have to dilute the saliva. 1 ml saliva and diluted with the 19 ml of the distilled water. Its ratio is 1:20 ratio. Then preparation of setup and experimental test tube setup. So all these things are required as in the procedure. So let's children, we have to understand now how to do. What is the procedure for the experiment? Again I am repeating, what is the experiment? To study the effect of different temperature on the activity of salivary amylase on the starch. So now we have to start this one. Basic requirement, once again I am informing this one, giving information to you. So we have the starch powder, children. This one powder is the white color of the powder. It's the starch powder. It is available in the laboratory. Similarly, sodium chloride NaCl. Then we require the potassium iodide for preparation of the iodine solution. And this one is the solution we have prepared. It's the freshly prepared iodine solution in the laboratory. Then distilled water, which we require. This is the distilled water. Now more things we require. These all are the setup which we require, such as the test tubes. We require the thermometer. We require the boiled water. We require the ice to maintain the very low temperature of the experiment. Then we require the test tube, funnel with the cotton filter is there for the collection of saliva. So we have to discuss now first procedure. What is the procedure? First, students, we have to prepare the starch solution. We have to prepare the starch solution. While preparing the starch solution, what to do? You have to measure the 1 gram of the starch powder. 1 gram of the starch powder. And this 1 gram of the starch powder, first you dissolve in 10 ml of the distilled water and prepare the one paste. So we have dissolved the 1 gram of the starch powder in 10 ml of the distilled water. And we have now prepared the paste. After making the paste, we have boiled the distilled water and we have taken the 90 ml of distilled water. So total water, boiled water is 90 ml. So total what? 1 gram of the starch and 100 ml of distilled water. And this solution is prepared. It is called 1% of the starch solution. Remember, 1 gram of the starch powder and 100 ml of the distilled water, and this distilled water was the boiled water, and we have dissolved the starch in it overnight. Then we have kept the overnight. And after the overnight, this one solution we have to filter. So already we have filtered. So for filter, what to do? This is the solution and this is the filter paper in the funnel. So we have to filter this solution. After keeping the overnight, we have to filter the solution. And after the filtration of the solution, now what we are getting? We are getting the 1 ml of the solution. So this is the procedure which we have done to obtain the 1 ml of the solution. So how to collect the 1 ml of the solution? So yes, let's we understand first. 1 gram of the starch powder and dissolve in the 100 ml of the distilled water. So that is the starch powder plus distilled water. So it is 1% of the starch solution. Similarly, we have to prepare the 1% of the NaCl solution. What we'll do? Take the 1 gram of the NaCl and dissolve in the 100 ml of the distilled water. Then third requirement is saliva. Dilute saliva is a third very important requirement for the experiment. And for the saliva collection, what to do? You have to take the one funnel, and in this funnel you have to make the one filter, and this filter is made by the cotton. We have to take the cotton, then this cotton we have to wet properly, and after this stick inside the funnel. So this I have made the filter from the cotton, and this now what to do? You have to take the chewing gum you have to take, or you have to take the rubber craft, and now grind with the mouth and collect the saliva. You put it here and collect the saliva. It means if you are taking the chewing gum, you chew it, and whatever saliva comes, that saliva pour inside this funnel. So we are getting this saliva. This is the saliva, and this saliva we have to mix with the distilled water. So here we have taken 1 ml of the saliva, and that saliva is dissolved in the 19 ml of the distilled water. So its ratio is 1:20. Now thus children, we have already now 1% of the starch solution. We have the 1% of the NaCl solution. This is the NaCl solution, and we have this diluted saliva. These are the basic requirement. Now second important part is, here you have to take the 3 test tube stand. 3 test tube stand we are having, and each test tube stand is having the 1 ml of iodine solution. All these 1 test tubes have the 1 ml of iodine solution. So thus in 3 test tube stand we have 6 test tube, 6 test tube, and this each test tube is having the iodine solution. Iodine solution you look like a pale yellow color. It's look the different pale. It's a yellow color, light yellow color it is there. So in test tube you set up 3 test tube stand. There are the 6 test tube in each, and each test tube we are having the iodine solution. Second, after this one we have to do the another important thing. One beaker you have to take the ice, and this is the thermometer where temperature is the less than here 0 degree temperature, or this is ice is the, so here temperature we have to maintain very low temperature. So maintaining the low temperature we have to take the, we have to take the thermometer to measure the low temperature, and this low temperature for experiment we have to take 5 degrees Celsius or more than 1-2 degree more. So 5 plus 1-2 or 5 minus 1-2. So here at least 5 degree Celsius temperature is maintained in this one. Another is because we are having the 37 degree temperature is there. 37 degree temperature is there. Third one, here it is we have taken the boiled water, and it is more than 50 degree temperature. More than 50 degree temperature. So in one beaker is the ice, temperature is low. Here it is 37 degree temperature, and here it is more than 50 degree temperature. Now children, what to do? You have to take the 3 boiling test tube. Boiling number test tube 1, boiling test tube 2, and what to do? First one we have to take in this test tube starch 1 solution already we have, and this starch solution 1% of starch solution prepared. So we have to take 5 ml of starch solution. 5 ml of starch solution. So it is more than 5 ml. So we have to take the 5 ml of starch solution. This is 5 ml of starch solution. I am keeping in the one test tube here. In this test tube also we have to keep the 5 ml. In this test tube also we have to take the 5 ml of starch solution. So in 3 test tubes all we have taken 5 ml of starch solution. Second, we have to take 1 ml of NaCl solution. 1 ml of NaCl solution. So I am taking the 1 ml. It is 1 ml. N ml is, I am keeping in this boiling test tube, 1 ml NaCl solution. In the same thing also we have to take 1 ml NaCl. Here also we have to take 1 ml NaCl. Already I have taken. Third important part is, this is the saliva, diluted saliva. And we have to take 4 ml of diluted saliva. 4 ml I am taking. This is 4 ml of the diluted saliva in this test tube. This 4 ml of diluted saliva, in this also we have to take, already we have taken, and in this also we have to take. It means children, in each test tube, what is there? In each test tube, in this first test tube, 5 ml of starch solution, 1 ml of NaCl solution, and 4 ml of the, it is the diluted saliva. It means enzymes is there. Now, after for boiling, we have to use the burner gas to heat the water and the for boiling, and this temperature now is more than 50° temperature. Here temperature again I have to see, this is temperature is 37° temperature, and here temperature we have to maintain then less than 5° temperature, or 5° temperature is required. So this is all set up and procedure. Okay, so now in this next one what we have to do? Next one we have to do, you have to take the separate droppers. This dropper we have to take. Okay, now 2 ml of, first one you see the iodine solution as I have mentioned, that is the pale yellowing color. So when iodine solution is pale yellowing color, okay, it's look the, now if I am mixing the, from this one, first one experiment, I have to take the little bit amount of this solution. And this solution we have to mix here in the iodine. So once I am mixing this in the iodine, so blue-black color comes. It's come the blue-black color. Blue-black color is coming. In this one, first experiment. Second, from this second test tube, we have to take some solution and we have to pour this one in this test tube. This test tube. Here also, if I am pouring, and we have to keep separate dropper and test. Here also, it's come the blue-black color. Blue-black. Then, in third experiment, I have to take this one solution in the dropper and pour this in this another test tube. Here, iodine solution is there. So here also we are watching. This is the solution is having the blue-black color. So, in our experiment, what to understand now? How to write, how to make the observation table, try to understand children. In procedure, as we have mentioned, procedure, preparation of 1% starch. How to prepare? 1 gram of the starch, and first dissolve in the 10 ml and then in 90 ml. So total 1 gram of the starch we have dissolved in the 100 ml of the distilled water, and we have kept overnight and then filtered, and this is the 1% of the starch solution. For 1% of the NaCl solution, we have taken the 1 gram of the NaCl and we have dissolved in the 100 ml of the distilled water. While preparing the collection of the saliva, we have collected the saliva and this saliva mixed with the water, and that ratio 1:20 ratio. And third one, we have also prepared fresh iodine solution in the lab. And this iodine solution we have prepared. Then what to do? Then we have done the experimental setup. So in experimental setup, 3 beakers we are having, and this 3 beaker, 1 beaker there is a ice, another beaker there is 30 temperature, and third beaker it is temperature is more than 50 degree because there is a boiling water. In the each test tube, this is having 5 ml of starch solution, 1 ml of NaCl solution, and 4 ml of the diluted saliva. Now we have to prepare observation. For observation, children, temperature column you make, temperature column. So the one place we have the 5 degree plus minus 2 degree temperature, at least 5 degree. Here second 37 degree temperature is maintained, second experimental test tube. And third we have 50 above temperature, 50 plus n minus 2. So this temperature we have taken. Now we have to see time taken or minute to reach to achromic point. It means what? What is achromic point? So children, with achromic point we have to understand that color of the, when we are pouring the solution, the iodine solution change into the blue-black color. But when saliva digests the starch, in such cases the blue-black color will not come. So that point where the blue-black color will not come in the iodine, and that point is said to be the achromic point. It means we have to measure the timing in which the iodine color remain as usual. Iodine color, as I have mentioned, this is the pale yellow in color. So when we are putting the starch is there, then they will give the blue-black color. But when no blue-black color come, it means the starch is completely digested by the salivary amylase. So we have to measure here time taken to achromic point. So for this one, as I have told to you, you have to take the this one solution. Once we have done this one, 2 ml of solution, and now after 2 minutes, again we have to repeat this experiment. Take again solution, and this solution pour in this test tube. This one also, it's a blue-black color comes, so achromic point is not reached. Second, again we have to take this one, and second test tube we have to mix the solution. Here also solution, the blue-black color comes, it means achromic point is not reached. Third one, from this one we have to take the solution and we have to pour in the other second test tube because it is after 2 minutes. Here I also mixing, here also this blue-black color comes. It means now starch is not digested. Children, this type now we have to repeat the experiment. One after 2 minutes, after 4 minutes, after 6 minutes, after 8 minutes, after 10 minutes, after 14, up to reach the achromic point. So up to this one we have to do the experiment where the change blue-black color will not come in the iodine. So in that case, if it is observed that our experiment for 5 degree temperature experiment is maintained, their achromic point is reached in 12 minutes. But 37 degree temperature is there, their achromic point, it means blue-black color will not come, iodine remain as usual color, and that reach to the 8 minute. As third experiment 50 degree temperature, we have received the achromic point. It means iodine color is as usual, starch blue-black color do not come, and that it means here is the 8 minute is the less time. It means 37 degree temperature we are receiving the achromic point at the 8 minute. It means salivary amylase is very active on the 37 degree temperature. This is the, but here if low temperature is there and high temperature is there. In such cases, in low temperature, enzyme get inactive, deactivated some enzyme, and high temperature the enzyme get denaturated, and that's why here more time is taken for the achromic point. So low temperature and high temperature, in this phase we are getting the more time for the to get the achromic point. But 37 degree temperature, there we are getting the achromic point at the 8 minutes. So this timing will be varies. So this is observation. So what to write in the observation? Temperature 5 degree, 37 degree, 50 degree plus minus 2. Time taken here 12 minutes comes, here 8 minutes come, and here 18 minutes. So less timing is consumed. So let's now we have to discuss about the explanation, children. Students, after the our observation, we came on the conclusion that the on different temperature 5, temperature 37 degree temperature, and temperature more than 50 degree Celsius. So in temperature 5, time taken, it means complete digestion of the start by the salivary amylase, it is 12 minutes. Whereas in 37 degree temperature, the time taken it's 8 minutes. And 50 degree temperature, the time taken for the digestion of the start by the salivary amylase, it is in the 18 minutes. And so that in study the effect of different temperature on the activity of salivary amylase on the start, what do we come to the conclusion? We come on the result. Number 1, 8 minutes are taken to reach the achromic point. At which temperature? At 37 degree temperature. 8 minute timing is taken. Because the enzymes is maximum active on 37 degree temperature. This is the very important point. The enzymes is maximum active on the 37 degree temperature. Then at low temperature it is 5 degree and high temperature is 50 degree. In these cases, time is taken to reach the achromic point is more. As we have 12 minute timing, it's more than the 8 minutes. And here also we have 18 minute timing as more than the 8 minutes. So in low temperature and the high temperature, the time is taken to reach the achromic point is the more. Now at all, on low temperature what happens? Actually enzymes are the proteinous in nature. So on the low temperature, the enzyme get inactivated. They get the inactivated. But in the high temperature, the enzyme get denaturated. And that's why more time is taken to reach the achromic point. In this experiment, basic requirement, precautions we have. So, precaution are filter the saliva with the cotton film, not by the filter paper. That's a very important. Second, all the wetting and measuring should be done very accurately. And third, all the glassware must be cleaned and dried. So thus we have came on the conclusion that how the salivary, how the different temperature is affecting on the activity of the salivary amylase.